Hello, everyone, and thank you for listening to today's podcast. What you see in front of you is a giant mammal that lived among humans some 10,000 years ago. Its name is the woolly mammoth. It had giant tusks, and its thick fur kept it protected from the harsh weather. The woolly mammoth eventually went extinct due to climate reasons and the fact that humans were hunting them as well. But now scientists are planning on bringing the woolly mammoth back to life. Some of you might be wondering, how can they bring something back from the dead? The answer is to plan on using a method called cloning to bring back the woolly mammoth. Take a minute to talk to your friends and neighbors and see what you know about cloning. Okay, so cloning is basically making a copy of an organism. But if you've never heard about how cloning is done, you'll be amazed how scientists actually do it. There are two main types of cloning methods: artificial embryo twinning and somatic cell nuclear transfer. Artificial embryo twinning is done by first getting a two-cell embryo. The two-cell embryo is then split in half and each cell is placed into its own petri dish. What happens is that the two cells will grow on their own even after separation. The two resulting cells are placed into a separate mother, and the two mothers will deliver the exact same organism. The two offsprings are clones of one another. Another method of cloning is somatic cell nuclear transfer. What happens first is the nucleus of an egg is removed. Then the nucleus of a somatic cell from an organism is removed and placed into the egg. A somatic cell is any cell other than a sperm or egg cell. Since the nucleus of the somatic cell carries a full set of chromosomes, it will develop into the exact same organism from where the somatic cell came from. Have you guys ever heard of the sheep dolly? She was the first successful mammal to be cloned using somatic cell nuclear transfer. The main scientist who plans on trying to clone the mammoth is named Professor Iritani. His plan is to use a well-preserved mammoth carcass in a Russian laboratory. His plan is to remove a somatic cell from the carcass that has undamaged DNA and plans on using the somatic nuclear cell transfer method. But let's take a second and think about this. So far, all cloning attempts have been used with living somatic cells. Professor Iritani plans on using a cell that has been dead for over 10,000 years. What makes somatic cell nuclear transfer possible is intact DNA. Once an organism dies, the DNA gets fragmented really easy because oxygen breaks it down. So the chances of Professor Iritani finding a cell with intact DNA appear really small. Take some time to discuss whether or not you think it's possible to bring back a mammoth. Why or why not? If Dr. Iritani can extract intact DNA, it will be placed inside of an adult African female elephant. If the pregnancy is successful and the baby elephant is born, it will not be considered a full mammoth. The baby elephant will be considered a hybrid between an elephant and a mammoth. This is where the moral issues about this process become involved. Cloned mammals are not known to live as long as real mammals, so the chances of it dying within a couple of years are very high. It has been seen with Dolly that many health issues arise that their bodies can't fight off. They live a short and painful life. Does this make it right to clone animals in the name of science, knowing that cloned animals will suffer? Another issue is the fact that if the cloning process is successful, what is there to stop them from continuously cloning hybrid mammoths? The fear is that if a clone dies rather quickly or goes unsuccessful, that they will just go and create another clone. These clones will be test subjects and will not be given the chance to live a normal life. What are some ways you think to prevent this from happening? So the real question is, will Iritani's cloning attempt be successful? Only time will tell. Thank you for tuning in for today's podcast. Hope you all have a great day.